focus on the AI, the digital transformation and the pathology applications. So Dr. Song is very experienced, and he's MD and a PhD, and right now he has a tremendous background training in the U.S. hospital and the universities, and right now he is started his own company called iCura. So today he will detail about his experience in AI application in these areas. So now to let you know, I don't talk too much. Just turn that to the Dr. Song and let him to give us a talk. Thank you very much, and Dr. Song. Right now, everything is on your hand. Song, Dr. Song, Dr. Song. Yeah, you can start it.
Looks like Dr. Song still have some technical issues.
Can you hear me now?
We can.
Okay, that's great. So I think I can share my screen from now on.
Yeah. You just let you start it.
Okay. So let me share the screen first. Please let me know if you can see my screen.
Yes.
Okay.
Very well. Very well.
So that's great. Hold on, let me see. Okay, everybody can see my screen and hear me, right?
Yeah.
Thank you all. I'm so sorry. There's some technical difficulties. I'm glad we actually resolved it. Hopefully, just a few minutes late. Thank you all for your attention. Oh, I have to apologize. My camera is not working either, so it's just going to be the presentation and me speaking here. Thanks for understanding. My topic would be digital transformation and AI in histopathology. First of all, I want to thank the organizer of this forum for inviting me, and it would be a tremendous opportunity for me to present my stuff and also learn from others. Let me start. So the content of this presentation will include mainly 5 sectors. First, I will review of the progress or history of histopathology in the medicine and drug development history. Secondly, I will present some current standard of care and ongoing digital transformation in the field of histopathology. Thirdly, I can discuss about some AI-enabled diagnostics and research tools, and these would be, I would say, the focus of today's topic. And then I will, from an end user point of view, I will try to present some challenges and potential opportunities in this particular niche area. And then I try to predict some future perspectives, and hopefully this will initiate some discussion between the AI and computer science part of the industry. And then the medical field, and this is definitely a cross-disciplinary area, and then collaboration is unavoidable. Histopathology, I'm not sure how many of the audiences here are very familiar with this, because I've taught, when I switched to industry after my hospital job, I talked to many people, and then it turns out there's a large percentage of the people who don't know about pathology or histopathology. Some people think it is all just about dissecting cadavers and giving a forensic diagnosis or something like that. However, histopathology has been a very important medical specialty, and it's developing and evolving along with other medical specialties, especially in terms of diagnosis, right? So it was considered that the founding father for modern pathology is Dr. Rudolf Virchow. And at the age of invention or advent of microscopy, Dr. Virchow was the one who started to focus on study at a cellular level of cell and tissue, level of tissues. So the first era of histopathology it's in early practice in 19th centuries. And then the foundation emerged in the 19th century, again, like I said, with the advent of light microscopy and some improved staining techniques. Dr. Virchow, known as the father, emphasized the cellular basis of disease, meaning that before that age, people can only see what they can see with bare eyes. But then once the microscope is available, we can start to see the tissue from the cellular tissue level. And from then on, believe it or not, until today, probably more than 100 years, 200 years maybe, hematoxylin and leucine stain, it's still the mainstay of this entire field in terms of histopathology. And it's considered the gold standard for tissue examination. Of course, I will explain how is that actually related to AI later. I'm just making this disclaimer all the time because I'm not 100% sure of the audience and all your interest areas are. Okay. Standardization of integration of histopathology into modern medicine starts in the 20th century, especially later 20th century, where histopathology became essential for diagnosing cancers, inflammatory disease, and infections. Technical enhancements, including the invention of paraffin embedding and microtomy, cutting the tissue into very thin sections, almost like under 5 microns. And there's the progress of automation in the histopathology lab. You start to have automatic tissue processor staining machine and stuff like that. And it was within the last couple of decades, and the specialty of pathology started to diverge into subspecialties. And these days, you could have general surgical pathology, cytopathology, hematopathology, dermatopathology, and all those kind of subspecialties. So shown here, it's an interesting image of a skin infected by fungus. It's probably Candida. And then you can see this is the very fundamental hematoxylin and leucine stain, and the fungus is shown there. And then why is pathology evolved into a full medical specialty where medical students graduate from med school? They're going to have to go through very long time of training, right? In the United States, there are residency system for either AP, CP, or AP-CP combined. AP meaning anatomical pathology, which is really focused on histopathology. The CP meaning clinical pathology, which focus on laboratory management as well as liquid-based test management interpretation. So histopathology mainly is in AP, but even for AP only, residency requires 3 years, and many residents will have to take another 1 or 2 years of fellowship to eventually become a independent attending pathologist. This is why I'm showing here on the screen. So what you're seeing is a cholangiocarcinoma metastasized to a lymph node. So you can see from the picture on the left side where there's this apparent some islands of cells or clusters of cells that differentiates themselves from the background of a little bit more even texture and color. But are these 3 islands the same, or are they the same thing? Probably from an untrained eye, it's hard to tell. And sometimes it's even hard to tell for trained pathologists. Where I want to say on the right side, it's a zoom in on these two circles on the left side. You can even zoom in at this level. This is 200x microscopy. And then by comparing the cells in this upper island or cluster to the lower island cluster, it's still very hard to grasp whether there's any differences in details and features. But there are, because for a trained pathologist, qualified, board-certified pathologist, the lower cluster is apparently a carcinoma, and the upper cluster is actually a very benign and normal proliferation center of the lymph node. The reason I'm showing this picture, because I'm trying to put some foundation for the later discussion about how AI or image analysis can be trained and how difficult they will be trained in terms of grasping all these details and identifying the differences. Again, this is another zoom in. This one is probably a 400x microscopy. Even at this level, some people probably start to see there's some difference in terms of every single cells in this cluster compared to every single cells in this cluster. Did anybody say something? Any questions? Okay. So that's why basically what I'm trying to present here is that you can appreciate that how much of a detail in between of a normal tissue or normal cell compared to a malignant or diseased cell. And that's where I think AI and digital pathology has to resolve, which actually has not been doing a very good job yet. So in this particular slide, I want to present the main ideas in terms of, for a human eye, how do you immediately or in a very short period of time identify abnormal cells or tissue compared with the normal ones, right? So there are two levels. One is overall pattern, meaning the organization of everything under the microscope. And this only takes you less than 1 second for a trained eye, for a pair of trained eyes to identify there's abnormality, which particularly refers to, does each tissue type respecting each other, right? Are they having a appropriate distance from each other, and are they well organized, and then not invading into each other's space? If you see something that apparently is disrespecting each other, meaning that their density, their distribution, their organization apparently showing something is invading into other things, places, or territory, and that's a trigger of suspecting an abnormal process, whether it's an infection or most likely it's a malignancy. So you can see upper two panels here. On the left side, it's a very normal breast tissue, right? Human breast tissue. You can see this island of breast gland cells by itself is a nicely smooth oval shape, and within it, you can see this tree-like distribution of the nice glands, and every one of them is maintained at almost equal distance from each other, not too much broader or narrower. So when you see this image, you immediately have a peace of mind, right? But on the right side, you start to see, and these cells are exactly the same cells that are shown in here, which are the cells lining the breast ducts and glands. And in the right side, apparently, they're not in any contained structure, not a contained oval island-like structure. They're everywhere, and their shape is all kinds of shape, right? Some of them look like a fish. Some of them look like whatever it is. And then they're haphazardly arranged among the background of fibrous tissue and collagen. So this is apparently a feature of malignancy. And the next level of features, we would zoom in to start looking at a cellular level. So for cells, you're mainly trying to capture first the nuclei. Is there any abnormality or deviation from normal nuclei of the cells you're looking at? Secondly, is the cytoplasm and the cell membrane, right? So for Again, the same thing on the left side on the bottom. It's a normal pancreatic duct. Every single cell is kind of lined up next to each other. They're not too overlapping or squeezing into each other. The distances between each cell is somehow maintained. And then the size of each cell is somehow maintained. As well as if you look at the nuclei, the little round shape in each cell, they're mostly oval or round. They're not giving you any weird shape. But compared to the right side, you start to see this is a pancreatic ductal carcinoma. So this is a carcinoma derived from normal tissue from the left side, right? So now you start to see within the so-called gland, the shape is very weird. You can tell some of them are forming a network. Some of them are having all these kinks and the abrupt changes, almost forming an angle. And these are definitely not the right structure. And if you look into the nuclei, so this one is a pretty big nuclei compared to someone here. So if you look at this small one and this big one, you know the small size is probably just 1/4 of the big nuclei. That's what we call anisocytosis, which is a feature for malignancy in pancreatic cancer. And then when you start to look into other cell nuclei.
Would you please point to the figure?
Oh, so the one in the left lower panel, right lower panel here. Do you see my mouse? You don't? Do you see my mouse, by the way?
No, we cannot see your mouse.
Oh, that's probably why. Okay, sorry. I thought you guys can.
We
sorry.
can see, but you have to move slow. Now I can see, right? But if you move
okay.
fast, we don't.
Get it. I'll make it slower. So, can you see this cell? My mouse is around. This black mouse.
Yes.
Okay. So again, let me repeat quickly. So this is a cell within a malignant gland of pancreatic ductal carcinoma. And then if you compare this nuclei to the nuclei here, so you can see this one is at least 4 times bigger than this nuclei. So that's what we call anisocytosis, meaning the nuclear size is variable. And then the nuclear size could be, some of them could be 3 or 4 times bigger than the other one. And that's just the size. And then when you start to look at the color, like the eusin stain, let me find my mouse. If you can see where my mouse is, there's a super dark nuclei here. And there's another one here. This is what we call chromatin condensation, hyperchromasia, meaning that the chromatin in this nuclei is heavily stained by hematoxylin, and that's another sign of malignancy. Okay, so I'll stop here in terms of trying to describe into the morphological differences. I think the main point I want to address here is that for people who are actually developing AI algorithms or models, trying to learn how to diagnose or differentiate normal tissue from malignant tissue, those are the features that needs to be kind of transferred to these people so that there is a better understanding how to develop the algorithm. So that's where my point is. I don't think that this one is has been very thoroughly communicated between the pathology field and the AI field. That's why we had a lot of discussions with potential collaborations with AI companies or teams, and we found out that that part is the hardest idea to be delivered, to be crossed over to the other field of specialty in AI, right? Okay. And then after all that, we talked about current days, most of the AI developing company in pathology are focusing on hematoxylin and eusin stain, the H&E stain images, just like these images. Basically, we call them pink and blue because that's essentially the color theme for these images. However, does AI need to go through all the H&E images or the textbooks over the last 100 years to learn pathology. I don't think anybody has a absolute answer yet. But from our perspective, or myself's thought process over the last probably 10 years, we think there's an alternative way of thinking how to deal with this. Especially when people started to build AI, they start with the so-called gold standard, because that's created by human. However, we all know that computer vision recognition can always recognize something human eyes can't. For example, what if we stain the tissue with just one stain, one color, and then just let the algorithm to identify all the differences in the shape, size, organization of these cells and tissues? Is that possible? My current answer is I don't know, but I'll talk about it later, that certain AI models are going that direction, which I think is very promising. The second trouble in terms of developing AI in pathology is the annotation effort and cost, right? For example, if you want to develop a supervised learning, then you're going to provide a lot of learning slides or learning images to the AI. But on those images, you have to tell the AI model, for example, what kind of shape or structure you consider normal, and what are the ones that you consider malignant, if your model is to tell malignancy from normal cellular tissue. There's many efforts in the past 5 or 6 years. We know there's some successful companies that had FDA cleared AI algorithm to tell cancer from normal tissue, but the cost was very, very heavy, because I know some company have hired more than 500 pathologists sitting there manually annotate each image they want to feed to the training model. Maybe more than 10,000 images, but for someone to manually annotate these images, just the one image could take you maybe at least 30 minutes to an hour. So you can imagine how big that cost is. And on the other hand, is unsupervised training possible? Meaning that you just keep feeding to the model with unannotated, not annotated images, but only tell the model the end result. Meaning you can feed 5,000 of slides to the model and say, these are all breast cancers. And then another 5,000 and telling the model that there's no cancer in these breast slides or whole slide images. I think there's also an effort going that direction, which we can probably touch a little bit later. So actually, just to make it a little bit more clear, I want to summarize what we talked about just now. We talked about the evolvement of pathology from all the way back, starting from 18th century until today. That H&E stain has been the mainstay and gold standard for diagnosis, no matter it's for cancer or it's for infections, and even other, say, metabolic disease like fatty liver disease. But then most of the AI models that are being developed, that were developed and still going on, are based on a belief that AI needs to learn pathology on H&E as well. So what we're proposing here is that we don't think that has to be the truth, because for computer vision recognition, there are a lot of ways. As long as you can have a way, for example, in this stain, this is toluidine blue, very cheap and simple and quick stain, probably much cheaper and quicker than even H&E stain. You can already show the different structures, shapes, and their organizations with each other. So this might actually be even better training material for the AI model. Okay, So let's move on. So when the time goes to late 20th century and early 21st century, especially starting from 1970s, for the staining part, in addition to hematoxylin and H&E, the one we just called pink and blue, immunohistochemistry was introduced. And this technology basically uses antibody to identify the antigens on different tissue, different cell surface markers, or sometimes cytoplasmic, like intracellular markers. And with certain enzyme-linked reactions, you can have a chromogenic color reaction and something turns blue or dark brown. Or alternatively, you can use fluorophores with horseradish protease, HRP, or any kind of other enzyme-linked reactions, so that you can see certain protein markers which has corresponding antibody to show up on a tissue section or on a histopathology section. So this has been proven to be a big leap for the histopathology world, both in diagnosis and research. And even today, probably every year, there are more and more IHC biomarkers being identified, some of them being introduced into clinical diagnostics and becoming FDA approved, right? Especially PD-L1 is a great example. We all know that Keytruda, all this and all this kind anti-PD-L1 or anti-PD-1 treatments came out after PD-L1 being a star biomarker in terms of promoting the immuno-oncology therapy. In addition, with the advancement of sequencing or PCR and sequencing technology, now when we walk into the early 21st century, next generation sequencing, digital PCR, which basically is a version of more advanced qPCR or RT-PCR, all this kind of technologies helped to give the researchers or even clinical pathologists more tool to get to know the molecular changes, not just the protein level, but now you're coming down to either DNA level or the transcribed mRNA level, or even other RNAs that are not functioning as transcriptions. So in this case, we start to learn more and more molecular and cellular level mechanisms of disease or normal processes. And this certainly helped a lot of new drug development as well, corresponding to the so-called disease-driving biomarkers. HER2 and PD-L1 being some very prominent example. And these ones eventually get developed into so-called companion diagnostics, because with testing of these markers, the usage of certain drugs are justified. This is an example of excellent IHC biomarkers. On the left side, you will see this is an image of invasive prostate carcinoma. And then what's staining very dark and brown in the nuclei, I hope you can see my mouse right now, very dark and brown, these are the nuclei of the prostate cancer cells. And this marker is called NKS 3.1. This one is very recently being used in the clinic. Back in the days I was doing my fellowship, this is probably 10 years ago, it was just started to be used in the clinical pathology labs. What it does is that very helpful if you're diagnosing a metastasis cancer from somewhere else into a different organ, where you start to see this the cells on this image where you don't know where they're from. And then you can use the NKX 3.1 to prove that they are from the prostate. The same thing on the right side. SOX10 is an excellent marker also for the nuclei of malignant melanoma cells. So melanoma cells has a very interesting character, where on the simple H&E stain, the hematoxylin, the blue and pink stain, they could look like anything. because different melanoma in different patients, they could have all kinds of morphologic features that are different from each other. Okay, sometimes even melanoma from the same patients on different sites will show different morphological features on just the H&E. So it used to be a very difficult disease to diagnose in terms of malignant or metastatic melanoma. However, over the last few decades, good markers like S100 and Melon A, HMB40, as well as this one shown here is called SOX10. They are able to successfully identify malignant melanoma cells, or actually they're also positive in normal benign melanocytes, so that you can easily identify or diagnose a melanoma when it's metastasized into a lymph node or even into brain or into your muscle, anywhere that they could metastasize into. Again, I want to stop here and reflect this discussion back to the main focus of today. IHC markers showing color different than hematoxylin and leucine, so they become also an interesting target for AI algorithm development. So that's the purpose here. Okay, so now we can talk about the most recent time period, which is after 2000, 2001. And there's a big surge in terms of so-called digital transformation. I believe back in 2011, 2012, this is almost 15 years ago, we started to see in hospitals, probably they came into academia research or industry research even earlier, we start to see hospitals that Apurio AT2, which is a very low throughput. You can scan probably only 2 or 4 slides at one time, and every scanning is probably taking 40 an hour, 60 minutes an hour, something like that. But they are the first models we started to use to scan so-called whole slide images, where you don't have to put the glass slides under the microscope to look at them. Now you can convert all of them into a digital image where it has all the advantages of a microscope and even more that the microscopes don't have, right? And then this is exactly the same reform that happened in the medical specialty of radiology, especially when digital camera, CCD and CMOS technology advanced over the last 30, 40 years. We all know that when if 20, 30 years ago, if we go to a clinic or hospital and get an X-ray or MRI, everything, they will give you a MRI is actually already digital, but X-ray would be a film. And then if you want to take that film with you, you can. And next time, if you go to another doctor to have a different opinion, you're going to have to present that film for that doctor to look at. But very quickly, about maybe 20 years ago, 30 years ago, the specialty radiology started to adopt digital imaging, just as everybody was taking photos when you go out to a tour or have a vacation. You have to purchase film, if someone here remember that. But very soon, in early 2000, digital camera replaced all this film camera. And the same thing happened in Department of Radiology. And 20 years later, probably in early 2015, around 2010, 2011, in the hospital, we start to have the digital whole slide scanner for those glass slides. And that's what we call the start of the digital transformation. And this simple transformation made everything possible, a lot of great things possible, including artificial intelligence, which actually came much later, maybe 5 or 10 years later than the digital whole slide image is available. And eventually it also evolves into, in the past maybe 4 or 5 years, something called spatial biology, where it purely depends on digital imaging in terms of wet lab stained images. So let's have a little summary again. So we discussed about the evolvement of histopathology in medicine for 4 stages probably, and then in each stage we discussed about the things that human eyes are trying to capture, and then we also proposed how computer visioning, like basically digital camera sensor, can convert it into images, and how computer algorithms could be developed. And those are all going to be the fundamentals for raw data for essentially for AI developments. We can switch gear now to discuss a little bit about drug development, because we know that this series about AI in drug development, and probably some of the audiences is wondering, what are you talking about? We're not trying to make diagnosis to patients. That's correct. So essentially, histopathology plays very critical role in drug development, especially when it gets to the later stage. Of course, probably not the compound screening and those kind of things. But when you started to get into preclinical studies on animals or evolve into clinical trials, in a lot of areas, histopathology plays very important role, as shown here in the table, right? Although the preclinical histopathology is not the main topic today, but as you can see, there's really not too much of a difference between animal histopathology from human histopathology. The only difference is that the organs and cells do look different a little bit, so you need specialty trained veterinarian pathologists to interpret those. But for developing AI, the idea and concepts are pretty much going to be the same. Okay, so now we want to specifically just give it 20 seconds to discuss about ADC, which is very hot and very advanced area of drug development. You can see that from target validation, safety, efficacy assessment, all the way to companion diagnostic, biomarker development, and even the post-treatment resistance and translational research. A lot of areas needs histopathology. So that's why as iCura is important, highly invested in this, in kind of a niche area, but we do believe there's a strong market need, and that there's a lot of values we can provide for the drug development community. Okay, so we talked about, I mean, the first section so far, we gave a brief introduction of histopathology in its history, and also how it's related to digital pathology, and how digital transformation, what's the concept of digital transformation in histopathology, and how they might be implicated in AI development. And now let's talk about what's the present status quo, how people are using digital pathology, how people are using AI in both diagnostics, like clinical diagnostics, as well as research, translational research, as well as even drug development. So here's some statistics here. There are more than 100,000 CLIA, so-called CLIA labs in the United States, right? And then the percentage of using or starting digital pathology right now, it's only 33%. And then fully adopted, which is only 10% out of the 32,000. Actually, these are probably CAP accredited labs. So what are the roadblocks that is preventing, since the technologies are all out there, you know, they're not really too expensive compared to the healthcare cost today, but they're just not easily and fastly being adopted by all the clinical labs, as well as translational labs and research labs. Of course, the first one is the cost. Some institutions, some labs are large enough to afford not only just the scanner, but also you need IT infrastructure, right? And then interoperability with the EHR, electronic health record, and LIS, laboratory information system, so that the digital pathology slides can flow through all these platforms relatively efficiently, which is not a cost that can be easily afforded by all sizes of labs, right? Secondly, it also depends on, especially in a clinical diagnostic lab, everything you use needs to be FDA approved. If not, you're going to have to go through very complex so-called validation to make a lab developed test called LDT. Again, not every lab has that capacity to do that. On the other hand, reimbursement is a different thing. We all know that most of people go to hospital, they should be covered either by their work insurance or they have to purchase private insurance. Anyhow, you just cannot pay everything out of pocket. However, when the insurance company pay the hospital or the physicians or the healthcare provider services, there are certain things called reimbursement rate, meaning you have to have a CPT code so that to justify yourself charging to the insurance. However, the CPT code part of digital pathology right now is still not a well-established system. So that's one of other reasons. The third thing is that that's what we were told when I was in a clinical informatics fellowship, that actually the most important resistance of digital transformation, probably in any industry, is the workforce resistance. Meaning that if you are, say, if a pathologist has gotten used to reading glass slides in his own office for the last 20 years, and all of a sudden you start to tell them that now we're going to put a screen, a big screen in your on your desk, and you have to read everything on the screen and use this mouse and keypads and everything. That's not an easy transition, as you may know, and you can easily hit a very a stiff wall and get an absolute no from the workforce. You can probably train the kids, I mean not kids, like the younger generation from med school and residency to get them accustomed to this, but it's gonna be very hard to try to convince the middle or older generation in their more advanced career years to switch something new like this. Okay, so that's the current standard, meaning although it is very strong and highly effective, efficient tool, there's not many labs in the current practice in the United States are using it. Okay. Okay. After we talked about the overall adoption of digital pathology, now I want to present a little fact about how even the current practice of pathology is doing on some of the diagnostic processes that might be completely out of your imagination. Because we know the medicine to this year's 2025, modern medicine is progressing at lightning speed every year. But I can tell you, in a pathology lab, if you're not using digital, or even if you're using digital, if we're counting mitotic figures, this is just an example. Hopefully now I'm trying to, this is my cursor. And then you start to see these kind of pink stain or dark blue, purple stain things. These are called chromatins. And all these chromatin congregation in condensed together, this is where the mitosis is happening. And we call this kind of observations in the cells the mitotic figure, okay? So why do we care about all these different mitotic figures? Because a highly mitotic cell is most likely to be a malignant cell. However, there are definitely exceptions, especially if this mitotic figure look ugly. Okay, we talked about how normal benign things should look beautiful and give you peace of mind, but malignant things look really haphazard and sometimes forgive me the language, and maybe disgusting to look at. Sorry. Like you see on the the little panels number from number 1 all the way to number 8. These are the mitosis that's supposed to be normally happening in cells, right? They're either a very round cluster or a club-shaped or two club-shaped. This is where they're separating, the two groups of chromosomes are separating, or at least forming a line or two separated dark lines like this. So this kind of feature tells you that they are most likely benign cells or tissue. And now you started to look at the bottom from 9 to 6. All these kind of mitosis are abnormal. First one is a very typical one, very classic. We call them in the clinic, we call them the Mercedes mitosis, where you can see there are 3 kind of little angles sticking out 3 different directions, almost like the Mercedes car emblem. That's why we call them Mercedes mitosis. And this one, you have one big blob of chromatin moving to one end and leaving a much smaller, disproportionately smaller part to the other end. But we know in normal mitosis, you should have a strict split of the 46 chromosomes into 23 each group and migrated to each side, right? So, and all these ugly ones suggested that these are malignant cells. And in certain tumor cells, we have to actually manually count how many of these mitotic figures. In those cases, it doesn't have to be malignant or benign, but the counting of the mitotic figures determines what kind of a grade of the cancer it is, especially important in neuroendocrine tumor. And in the current day US labs, many pathologists spend hours every day to do manual counting, looking at a glass slide and a microscope, and then on the other hand, with a pencil, writing down the number 1, 2, 3, 4. I don't know if that shocked you, but it does shock me. It shocked me when I was getting this training in residency. It is still shocking me every day because this is 10 years past my residency, and it's still the common practice every day in the pathology lab. This is a HER2/neu. It's the companion diagnostic, first established by Roche with their Herceptin treatment on breast cancer, right? So what this is an IHC. Basically, you're looking at the brown color stain on the cell membrane. And then with this, based on this, you give this breast cancer a HER2/neu score, right? Meaning is 1+, 2+, or 3+. Based on these scores, you will qualify the patients for either the HER2/neu Herceptin treatment or not qualified for the Herceptin treatment. And why there's this qualification? Of course, there's scientific reasons. There's also financial reasons. Scientific or medical reason is very simple. Like if this patient breast cancer has a lot of HER2 receptor, a HER2/neu, and then the patient receives Herceptin, there's a much higher rate that patient's going to respond to that treatment and get better soon or get a much better prognosis. But if this patient the cancer does not express HER2/neu, and that means this treatment most likely is not going to be helpful for this patient. And then at the same time, if the patient took it, he or she will actually suffer from side effects. And that's the medical reason. And financial reason is that insurance company, they don't want to pay every breast cancer patient with this Herceptin treatment. They want you to have a differentiation justification for using this drug. So that's basically what it is, the essential idea. Okay, so again, stay here. Current day, I would say more than 70-80% or even more of those hospital labs, the pathologists doing diagnosis on breast cancers, they are using their eyes. They're eyeballing the so-called HER2/neu expression pattern and the scoring. Again, that broke my heart, but it's still the current standard of care, So why don't we, for such an easy, for me, I would say it's an easy algorithm development. Say, why can't we just have computer recognize all this darker brown or lighter brown lines around these cells and give a score that's more objective? Not only that, how many cells do you think a pathologist can count or give a percentage on a daily basis? You don't have the whole day for one single patient's diagnosis. Normally, a pathology has to diagnose at least 30 to 40 cases a day within the hours they're at work, right? So you can't spend a whole day on the simple HER2/neu calculation. And then the end result is what? The end result is that the inter-observer variances sometimes could be very high, meaning that the same case coming to pathologist A, he or she probably say, this is a 3+ or 2+, patient go get the Herceptin. And the next day you bring it to a pathologist B, and he or she will say, oh, this is a 1+, which means negative. The patient is not qualified for Herceptin. How can you prevent that? I mean, it's a very controversial topic, right? It's very hard to prevent that. I just put it this way. The same thing actually happens to ER, PR, HER2. There's regulations to kind of trying to tightly control that inter-observer variances, meaning there's so-called proficiency testing from CAP that everybody has to pass it, but okay, I'm not gonna get into too much details in that. Okay, now, so ever since 2012, I had my first exposure to so-called digital pathology. I started to think that there's a lot of things human can do much better than computer, right? But I personally believe computer also has its advantages, meaning calculation or anything that could be mathematically figured out. I would always say machines probably are much more accurate and consistent than a human, even mathematician. So ever since then until today, there are actually quite a few so-called image analysis algorithms approved by FDA as listed here. So you can see ER meaning estrogen receptor. Again, it's related to breast cancer. PR is progesterone receptor. All these three markers are tightly related to breast cancer. And breast cancer appears to be one of the major cancer type that get heavily studied by this predictive markers. And then later, HER2 actually got identified to be present in many other cancer types. For example, gastrointestinal cancers and stuff like that. But anyway, coming back to the focus of this topic, so far, these are the only few, probably, maybe missing one or two, I don't know, but there's not many algorithms on a computer that are FDA cleared. And you can see the lower two actually are not even FDA, they're RUO, meaning research use only. You can't use them on patient samples. This is just how slow we're developing in this front line. And then in terms of whole slide imager, like I said, back in early 2000, we were using, what did I do? Sorry. We were using the so-called ScanScope AT. You can probably put 2 slides or 4 slides in one time, and scanning that 4 slides probably going to take you anywhere between 20 minutes to maybe 30 minutes, half hour. And then I think these part of technology really evolved very excitingly. And these days, the newest generation of scanners, the same company, Aperio GT450, you can scan 450 slides in one run. And for each slide, the scanning speed can be as short as 20 seconds. So that's a really big progress and leap in that. And so is some of the other platforms that are shown on the right side of the slide. The top one is the Philips IntelliSight. That's actually the first FDA approved digital whole slide scanner. I believe it was in 2017, something like that. Following that, you have the NanoZoomer 360DX, I believe, S360DX. And then you have the Ventana DP200 from Roche. These are FDA-approved whole slide imagers to scan clinical diagnostic slides. And they are heavily also used by pharma for drug development, which needs a lot of the digital imaging tests. Okay, this is just an example. I think I'm running too much of time right now. I'll pull up like a little quicker. This is another example of current practice in terms of counting the FISH, fluorescent in situ hybridization for HER2 testing. You can see many labs are using their eyes literally under the microscope to count the centromere CEP17, chromosome 17 centromere, and the signal for the HER2 to judge whether to get to a ratio to judge whether they're amplified or not on the left side is definitely amplified. You can see within one single cell, you see 2 central mirrors, but so many more copies rather than 2 of HER2. But then in the right side, the normal cell should only have 2 central mirrors, 2, or maybe sometimes there's artifacts, 2 more to the HER2 sample. And then normally the guideline, the CAP guideline, College of American Pathologists guideline, is that you manually count 20 independent cells and come to a conclusion. But now we can do thousands of cells to get a much more accurate conclusion. So that's where we can segue into discussing AI-enabled diagnostic and research tools. Showing here is actually AI-enabled cell and tissue segmentation, where all this yellow and light white colored area, almost like a map, those are considered stroma, or the benign area of the tissue, and all this red marked circled areas are the tumor cells. These can be achieved by AI. And I'm probably gonna skip a little bit on the multiplex immunofluorescence, maybe give it a 30 seconds. So what this thing means is that it's another leap in terms of wet lab chemical staining advancements over the traditional H&E or the single brown color immunohistochemistry. Now, on the same slide, you can stain multiple markers with different colors so that you can see each and every single cell, whether they're expressing only just one marker or they're co-expressing different markers. And as many of you, definitely, I trust you guys have biology and medical physiology background. You know how exciting and important this could be. And this is an example of a Hodgkin cell lymphoma, where when you diagnose it in a pathology lab, you have to see at least 3, 4, or 4 markers expressed on the same cell. But it's normally impossible to do that because you have to do serial sections. Each IHC can only give you 1 marker. But serial sections does not guarantee that when you see the CD30 staining the cytoplasm on certain cells in your first section, when you see the second section looking for the same cells expressing MOM1 in the nuclei, you can't find all the exact location of the cells because these are not the same section anymore, right? So multiplexing has that power. And I listed quite a few other powers of multiplexing immunofluorescence or immunohistochemistry can do. This has been at the forefront today for many pharma and drug development biotech companies. And they're using it in variable different use cases. We might be able to present a few more. So the top 5 applications, including immunoprofiling of tumor microenvironments and the prediction for a treatment response, co-expression of CDX markers, drug screening for immunotherapies, and all that. And then the last one I talked about, AI-powered robust algorithms. Because guess what? Now you resolve one of the major headaches of AI model training, which is annotation, right? Mostly done by human manual annotation of images. Now with these marker expression, you can do AI annotations. This is just an example for a immuno checkpoint inhibitor drug developer, and we worked with them to look at the tumor microenvironments, where you can see all those different markers represents activated T cells, T helper cells, cytotoxic T cells, macrophages, their distribution among the tumor. And you can see this green CD8 positive cytotoxic T cell, they're already invading into the tumor tissue, starting to eat them. So this is a good sign for an immune hot tumor in terms of immune treatment, immune modulating treatment. This is just a reiteration of the Hodgkin's lymphoma diagnostic panel. And this next one is funny. This is a client who want to see whether their drug is hitting the target at the tissue level. So we developed a so-called tissue receptor occupancy assay, where there's an antibody against their drug and there's an antibody against the receptor. So you can see we can put the both staining with different color on muscle tissue. They're studying some muscle disease to show them that there is, by the dose elevation, there is a higher receptor occupancy of the particular compound they're looking for. In the same topic, when you try to look at whether your drug hits the right target, which is also a very important topic in drug development, right? A lot of drugs get rejected or failed at phase 2 or phase 3 with adverse effect because the drug is not hitting the right target. They're hitting other organs, sometimes are vital organs like hearts, like eyes. Some drugs could be targeting kidney and liver, but they go all the way to eyes and got blinded patients having blindness. And those are big issue. I think that's just because that the tissue distribution and on-target and off-target distribution has not been studied thoroughly. So with that issue going on and push the clinical trial further, it's inevitable you're going to have some severe side effects and adverse effects being found out, unfortunately, on the patient subjects. So what you can see here is that with this multiplex immunofluorescence, we can actually identify, especially on the right upper panel where my cursor is, this is a mouse. This is a mouse kidney. So you can see that with the specific marker for each different cell types, we can even monitor the drug distribution at the cellular level, meaning that are they hitting the podocytes? Are they hitting the endothelial cells in the glomeruli or the small vessels in the kidney? Or are they hitting the proximal tubule or distal tubule of the kidney? And once you have your fluorescent tag, the drug injected in the mouse, and you can get all those answers, all those questions answered. At the same time, if you are curious about the off-target distribution of your drug, and then you can check, you can check pretty much every single organ of that particular mice or group of mice to figure out whether your drug has a desired target distribution or it has a super off-target distribution where a side effect could be more than likely going to happen. And on this left lower panel is a liver section where, again, all these different colors represents different cell populations, including hepatocytes, Kupffer cells, bile duct epithelium, as well as endothelium of the blood vessels. If you have your fluorescence tag drugs distributing in there, you can see them as well. And on the right lower panel, that's a muscle. That's the same study we showed before. So I think try to save some time. I'll stop here on the multiplex immunofluorescence image. And then guess what? For at this time of the point, some of these manufacturers already have AI-enabled software that can go in and take all this multiplex immunofluorescence digital images. These are whole slide images. And then go in there and have the AI to do the so-called tissue and cell segmentation. What that means is that if you see this image, hope you can see my cursor here, show this image on the left upper panel, where all this blue and red are artificial labels applied by this AI-enabled software, where it tells you that which either color will be representing a certain cell type. This yellow is a different cell type, and then what are their distribution and then their borders with each other. Basically, this is what people like to ask in terms of immunotherapy development. They want to know first whether their drug is activating the innate immune system, where there's an increase of cytotoxic T cells, T helper cells, or activated T cells in the tumor environment compared to baseline before treatment, or whether this activated immune cells are getting closer or even invading into the tumor area to start the process of destroying them, right? So this is essentially the concept, so-called spatial biology. And this is on the protein level. Of course, you want to combine the transcriptomics level, where there's a 10x or nanostring platform where you can study the RNA expression level of certain markers or actual even cytokines you're interested in terms of their expression level in cells and their geographic relation to the tumor. So this has been very informative and greatly helpful for this immunomodulating drug developer or pharmas. And then on the right side, this is basically the AI-enabled automatic labeling of the cells and start calculating their geographic relationships with each other. And the data output is all quantitative. And then all this quantitative assay is based on thousands or tens of thousands cells in that area, where for human, impossible, right? You can't have someone sitting there and start counting thousands or tens of thousands cells in that area and start making all those mathematical calculations. So these has been a very powerful tool, and we have used this to support many of this drug development process. And this is a software actually open source, free to use from NIH. We've been using this for quite a long time. It's also a AI-enabled cell and tissue segmentation, having a lot of strong, powerful functionalities, but I'll just not go into the details. Again, like I said, currently, most of the labs are counting the fish probes by hand, and then you only can probably count 20 to 30 cells and stop there and give a conclusion whether this is a HER2 amplified or not amplified. But with computer algorithms, you can do thousands. This is what we did. This is, of course, not a breast cancer. So also the target marker, the marker is called target because I cannot reveal the client data. But we can use the AI-enabled computer algorithm to identify benign stroma, which is yellow labeled, and cancer cells distribution. And going into the field, you can see in this particular stain, the yellow is the centromere marker, and the red is the target marker. And you can see the cancer cells are obviously many, many more red compared to the number of yellow. And this is where the AI-enabled algorithm can go in there and count every single red dot. And the result you can compare. So the manual, probably our scientists did maybe 30, 50 cells. That's it. It's going to take a lot of time. But for the automated, we started out 3,000 cells. And then if you want the computer run a little bit longer, within about 30 minutes, you can count more than 10,000 cells, and you can compare the results. Although in this case, it's very similar, which is validate that the computer algorithm is correct. But in other cases, maybe the automated is different than the manual. You'll find out that the more cell you count, you're closer to the truth, and the manual counting has a disadvantage in those scenarios. Okay, so just also a little summary here. Basically, what digitization of histopathology did is completely transformed the field from a manual analog process into a quantifiable science in a digital sense, right? Infusing more and more objectivity into the subjective diagnostic tool. That's basically our understanding. Challenges and opportunities. First of all, we have seen many companies now trying to develop AI models for histopathology. One big confusion is that many people consider AI, that we talk about DeepSeek, we talk about ChatGPT, Gemini and stuff like that. But those are probably not the same AI that histopathology or even the radiology AI is going to need. Because for image recognition, these days, there are different platforms to handle different kind of things, which are listed here. Convoluted neural network is very often, but there are more. I'm not an AI expert, right? But listed here, there are more other, basically the fundamental models to build this kind of AI algorithm. Listed here. I'm not going to get too deep. I don't claim to be an expert here, but I know most of them what they're talking about, especially for their use cases, right? Some of them are really good on tissue segmentation. Some of them actually can do even classification, and some of them can do unsupervised, unlabeled data training. So that's what we discussed a little bit earlier. But what are the challenges for these companies, right? So for digital pathology, you can briefly, it's the mainstream adoption is the rate limiting step. We have already discussed about this. It's cost, it's reimbursement, it's compliance approval. and all that, right? But for AI, what are the major challenges? I would say, and I believe a lot of the people working in this field are talking the same thing. Access to standardized, high quality data is super limited, right? So if you're training a automatic driving AI, and then you can go out on the street, you drive your little car, little van, and put a camera on the top. You can take as many images of trees, roads, other cars, and then you go back, collect all this data to train your AI, because there's nobody going to charge you for taking those data, right? The same thing as facial recognition AI. You can put a a monitor camera in an airport or in a store. It's a public area, so most of them is legally allowed to take security or videos, whatever, and you can collect all those data. So in those kind of AI field, access to data doesn't seem to be a big hurdle or roadblock. But in regulated, especially healthcare, right? How can you have access to thousands and tens of thousands of different breast cancer specimens or digital images with all the regulations, with all the tight provided health information protection laws, such as HIPAA, and in the Europe it's called GDPR, and then with all this kind of regulations, and now it's almost impossible, especially for smaller operation companies on AI. Your first question would ask, you could have like all the talents from MIT or Pittsburgh, Carnegie Mellon for AI, but where's your data? That's the biggest hurdle. I can't get into details because there's a lot of perspectives, but that's the major problem preventing a lot of AI company in the histopathology to evolve, even in the healthcare, because accessing patient EHR is another issue, highly regulated, right? And then secondly, we already touched upon a little bit. So on the right, sorry, I want to talk about these images because highly standardized quality data, and quality is also important because a lot of data you might have access to in the hospital, they degrade it. They're not as good. So garbage in, garbage out. We all know that. So this is a nicely done H&E stain on the left side, and it's a poor one on the right side. How do you think that your model, when getting trained, can differentiate what's the nice data and then what's bad data, and try to pick up only the nice data, learn on it, and disregard the bad data? It's probably not going to be that easy to be achieved, right? Annotation efficiency and cost. I remember there was a company recently, maybe a young kid from Pittsburgh, sold a company for a huge amount of money just because he prevented annotated data. And then the model is that he went to Philippines or wherever, have the relatively more affordable, let's just put it this way, annotators for the data. But again, we can tell that in the AI training, annotation is still huge cost, right? Sometimes cost prohibitive for developing any AI. Accuracy and dependability, this has been discussed tremendously for people and users, right? Starting to use AI, even people using, let's say, ChatGPT or DeepSeek or Gemini. You can get the same result for the same question, even it's a mathematical question from two different large language models. So that starts to make people question. I mean, myself tried them myself, and I found out there's a lot of concerns about accuracy and dependability. And also another big thing is about explainable and transparency, especially in healthcare. Like people, if the AI tells me that this is a prostate cancer instead of just a benign hypertrophic prostate, why? Most of AIs, especially the CNNs, they are known to be black box. They are not going to be able to tell you. So how do you know that the AI is not just by luck had the right answer this time? But you want to apply, if this is a mature product, you want to apply to hundreds of thousands of patients, right? But if you don't know how the AI came up with the conclusion that particular this one is malignant, not a benign one, and you still have a doubt, that's a problem. So there are another group of people starting to talk about explainable and transparent AI, meaning that even the AI did most of the work, human can still understand how the AI kind of inferred or come to that conclusion, which is also another very difficult area to conquer. Again, at the end, who's the gold standard and ground truth? Is it pathologist? But pathologists are known to have disagreements. For the same thing, different pathology will tell you different opinion and diagnosis. How do you deal with that? Right? That's, I don't know. I don't know the answer for that. That's also an unanswered question. And there's used a famous saying from Dr. Rosai, who is considered one of the most prominent pathology in modern days. There's a story that he seated with 17 other pathologists in a meeting talking about melanoma, and then nobody knows the answer to a case. He said that's a melanoma, but all the other 17 says, we don't think so, we don't know, and we don't think so. How do you know that's a melanoma? And he said it's melanoma. After a few years, of course, the clinical outcome came back, the patient's outcome, it is in the melanoma. And then people were all amazed, and then came back. Dr. Rosai say, how did you know that? And Dr. Rosai said, it's hard to describe. It's just like when my grandma is walking from a distance to me, even though I could not see her face clearly, I know that's my grandma. But how can I tell someone standing next to me say, oh, that's my grandma? And the other one would say, I can't see it. I can't even see the face. And that's basically a joke going around. But sounds like, I mean, I think it was believed to be a truth. So that also tells you, how can you have human pathologists being the gold standard, where even themselves do not agree with each other? So that's another big question mark as a challenge. Okay, so opportunities, right? So these are some of, and this only represents my personal opinion. Domain-specific models, I think, is much more easier than foundational or general models, especially in image recognition, because different cancers and even different organs, the benign and malignant, look differently. So something might look benign in an organ, and then at that particular morphology, exact same kind of features would definitely be malignant in a different organ. So it's more likely you're going to have to start from designing models at least per organ system, if not per disease entity, right? Even breast, you has most common ductal carcinoma and lobular carcinoma, and then a few others, right? Your model for identifying ductal carcinoma is probably not going to work for identifying lobular carcinoma. The same thing applies for kidney, liver. Well, liver is easier, a little bit easier, because most likely it's just a hepatocellular carcinoma or cholangiocarcinoma, but it doesn't matter. So for you to have more accurate, dependable AI, it's more likely going to work out faster and less costly if you want to design it on a particular indication. That's why Page AI's prostate diagnostic algorithm just got approved by FDA. I believe it's 2024, right? You can show that later. Data format standardization. Most of digital scanners now can somewhat be compatible with other file image management system, but most of them, for probably various reasons, they maintain their specific data format, which is a headache, right? Other stains, we're running out of time, but I'll just quickly go through these, probably not get into too much of details. Future perspective, we probably already talked a lot about that. So I think WSI will be expanding from archival research to routine primary diagnosis. That's probably an unavoidable trend. Just like even just 4 or 5 years ago, a lot of pathologists are doing human transcription, meaning that they will talk to a tape recorder, and the recorded diagnosis will be sent to the office for the transcriber to type into the actual pathology report and send back to the pathologist, sign off, right? But then 5 years ago, a lot of hospitals started using voice recognition software. Believe it or not, there's always love and hate, different opinions about it, but it's already the mainstream. Digital pathology is not yet, but I believe it's gonna be maybe like the next 3, 4, 5 years. Hardware cost is still still kind of high, and then regulatory approval is still not so many, but probably we're expecting more. again, we talked about standardization of data format. Now you have SVS, MRSX, you have TIFF, you have JPEG, all kinds of different formats, and they have to be consistently standardized. People are talking about DICOM, which is the radiology imaging format. It's going that way. Probably everybody's going to go be conforming to that standard. Hardware as well. We know that photographers, especially people who likes to take photos, they know the differences between the pictures of a Canon, or a Fuji camera or a Sony. They all look different in terms of the tones or contrast, color, vibration, all those kind of things. So that's the same thing with scanner, right? So with the same slide scanned under different scanners and you show up on the same computer display, they look different. So how do we resolve that? That needs to be standardized. The same thing with computer display. Are you going to use a LG, or are you going to use a Dell or HP 27 4K, or it's just going to be a 22, 24 1080p? They all make a difference in terms of final image showing up on your eyes on the screen, and whether those differences going to have a significant impact on your judgment has to be validated. We don't know. And there are certain medical grade display companies having medical grade FDA approved pathology displays. Very few. I only know one, right? Okay, so there are already FDA approved AI, pre-AI or AI assisted pathology diagnosis. What you're seeing on the image here is a pap smear, which we know that since the pap smear has been widely adopted, the cervical cancer rate has drastically decreased. This is a very great advancement in diagnosis especially for gynecological cervical cancer patients or populations who do not have that anymore, right? And then these are very nice progresses, and then they have been proven to be very successful in actual clinical performance and everything. One day, I mean, probably pretty soon, we'll see something like this being completely FDA cleared, can be used for clinical diagnosis as well as translational research, and even biotech and pharma projects and studies. Okay, these are also some of the FDA cleared things on what we discussed about quantifying the markers, IHC markers, ERPR, HER2, and just to put this as an example for us to see and think about. I believe there's going to be more and more coming out. With all the difficulties, there's always hope, and then I think significant and solid progresses have been made, even within the last 2 or 3 years. Multi-omics is something we also strongly believe in, and this image you're seeing here is a multiplex immunofluorescence where it shows you the yellow is the pan-CK showing the invasive colorectal cancer, and then the green is a certain marker that makes them different. And then you start to see the red, orange, and everything. Those are the immune cells. Some of them are fibroblasts associated with cancer, meaning CAPE, cancer-associated fibroblasts. Some others are regular fibroblasts. These are all the hot topics in especially extracellular matrix modulating agents or drugs research, or immune checkpoint blockers or immune modulating drug development. I believe in the audience who are in this field probably will have more impression on this particular presentation. Other considerations mostly are in the clinical diagnostic field. But again, we talked about not only the training, validation, and human oversight. This is probably the ethical part of it, or how do we make sure it's actually working correctly? I just give you a very interesting example. I didn't know this until I started learning pathology or started getting into the residency, that for Not to mention that different diseases have different presentations or different prevalence in different ethnic groups or racial groups, even simply just the hairs, right? They call this Asian hair and this Caucasian hair and this was Afro. Actually, I don't believe that's really related to the race. Actually, but it's probably predominant for Asian people. The hair look like round, pretty round, and then relatively thicker, right? And for the predominant Caucasian hair, it's more flat, more oval, and then thinner, overall thinner. And this Afro textured hair is more even flatter, and they're much wider. And then they tend to be always curly, and these ones tend to be always straight. But I think that it just depends on every individual. As long as if you touch your hair, you feel like it's very soft and kind of thin, and you're more likely in this category in terms of your hair structure on histology. But if you touch your hair and feels like very rough, right? And then they never really come down to flat on your scalp. That means your hair is more toward this extreme of hair texture, and the rest of people goes into this category. And the example I want to put this in is that so why FDA wants a wide demographic variation in their final approval of a clinical trial or data. I think it's just because even every different racial or ethnic groups on cellular histology level, things might look different. So that's another layer of consideration. How you can collect large enough training data, right? For example, you're trying to develop something for ductal carcinoma, breast ductal carcinoma, and you probably want to include all the potential racial ethnic group samples where you can have that algorithm correctly deal with the potential future use cases, right? You can't just say, for example, you can't just collect like 10,000, another 3,000, everything from Asian countries and develop your AI. And then you come to FDA, say, I want approval. And then FDA approved it. And it turns out that AI is going to be used in US where the variety of variances of ethnic and racial groups are much wider in East Asia. And then you might run into issue there, right? So those are the actual realistic considerations that we can. There's a lot of discussions about this. I'm just probably touching the very tiny surface of it. It's just too much of a big topic, even from. I mean, I'm still a very initial learner, so these are just representing my own opinion. So in the long term, I think digital pathology AI will become the standard of care, although it's not yet. It enables high throughput quantitative data analysis to improve both diagnostic and drug development, any kind of translational research. And then I have no doubt it will help tremendously new tissue biomarkers and insights, supports global collaboration. Of course, now if it's digital, you can literally send it to every single corner of the world. People can collaborate on the same image from all over the places. I think that's all I have. Thank you so much. Hello.
Thank you very much for Dr. Song's tremendous talk. I think that's a really great uh, lesson for the pathology area. So that give the start from the beginning to the recent development, digitization or transformation. All these things is a very, very good materials. I think everyone will be very beneficial after your talk. So let give the just representing the whole organization and to appreciate Dr. Song's tonight's talk. This is a great talk. Thank you so much.
Thank
And
you.
now we start to open the discussion session. So you can type in your questions, and you also can open your uh, the audio. You don't need to mute that. You can open to talk. So first the question, let me start from the audience. The people ask you, say, could you explain annotated image and the typical H&E image with a diagnostic? I think the questions ask, what is these two type of imaging difference? Could you explain that? This
Yes.
came from the audience.
Yes. So annotated images meaning that you have a whole slide scanning, for example, of a breast cancer tissue, right? That itself is just a regular H&E image. But annotated meaning someone, pathologist going in there, can circle where the cancer is, right? And where the benign duct is, benign cells are, or immune cells are, or benign stromal tissue are. So that's called annotation. After you annotated these images, and then this image will be useful for training your AI. The AI needs to know what do you think they are to learn. So that's basically called annotated images. Hope that answered your question.
Yeah, that's good. So, is there any other question to ask?
This is Yang Wang. I have a question for
Yeah.
Dr. Song.
Sure.
Regarding the clinical application, and then currently, in the clinical lab,
Yeah.
the use the information system. Do you think this the pathology AI assistant pathology, when they capture so many the images and then get so many data, so the current the information system still can be used or need to be improved, or what is the current status?
You mean the laboratory information system, whether this needs to be improved to match with the big data that's generated by digital pathology?
Yeah, correct.
Okay, so that's a great question. I think that's also a well-recognized issue in the field. There are many newer generation laboratory information system that are born to deal with this problem. First of all, your IT infrastructure needs to be either completely overhauled or have a significant improvements in terms of managing large data, either storage or transit, right? Internal data transfer and stuff like that. So but mostly in pathology, they are developing a separate thing called IMS. IMS meaning image management system, right? There are mature products on the market already. The most well-known one is called a Sectra, SECTRA. It's a high throughput, large scale clinical diagnostic image and image metadata management system. Started out from Sweden. I think it's taken over 70% of the market in Europe, and it's growing in the United States. Other than that, there's several other companies in the US which are quite new and recent, but they're expanding very fast as well to take over the market. And then there was another one called, I don't know if you guys heard about Epic. Epic is the EMR, electronic medical record system, that's taking over at least 2/3 of the major hospitals information system. And they are also working on something like that to manage large image data from both radiology and pathology. I hope that answers the question. So I guess the purpose is that you want to know whether there's a certain opportunity to like business opportunity. I would say it's a complex complex judgment, but I think there certainly should be.
Yes, you read my mind. Thank you.
No problem.
Dr. Song, I have a question.
Sure.
The current for the drug development and also for the oncology part, and the endpoint to measure the endpoint and also to there's a standard is RACIST 2.0.
Mhm.
So that is you measure every step at a certain time period. You measure the cancer size change
Correct.
based on the imaging. But you also mentioned that the pathology imaging also can replace the also can be used as a endpoint to measure the process of the change of the size of the tumor.
Yes.
Is that right now is FDA officially acceptable or still in the research part or in which stage of that
I think
application?
right now it's not required by FDA in
Okay.
terms of the RACIST standard, right? Because
Mhm.
that's more like a clinical evaluation of the treatment response, or any changes of patient status. However, we have been working with several clients, and then we know that some of these new techniques or technologies that will be kind of required by FDA in terms of at least right now it's additional data to facilitate the drug approval with a strong evidence advocacy. Because, for example, FDA is trying to look at, say, the efficacy result, efficacy report, meaning that if you're trying to do an immunocheckpoint blocker drug, and then your belief is that the drug activates the immune cells so that turning a immune cold tumor into an immune hot tumor, and then you better have evidence to support that. And that's where we can use the pathology studies with the baseline biopsy or the surgical specimen testing for the immune microenvironment, and then have a post-treatment biopsy if there's residual tumor or metastasites in the patient's body to compare with the immune microenvironment to show the evidence that the drug actually played a difference in terms of activating the immune system to kill the tumor cells.
That's good. That's good. Thank you.
Sure. Thank you.
Anyone has any more questions?
Hi, this is Jia. I have two questions, actually a couple of questions. And first of all, a really nice talk. Thank you. I work with animal studies most of the time, so work with a veterinarian pathologist. But I guess there are similarities. So one thing I noticed you mentioned there, like when AI made a diagnosis and there's no way, or we don't know how it reached the conclusion. I'm just wondering whether it's possible to ask AI to provide a rationale for their conclusion.
No, I don't think so, because when we say we ask AI, we're still probably thinking about in the context of large language models, where because these models are dealing with managing the semantics of words, sentences, and everything, and the logical thinking is based on what the message is delivered, right?
Mm-hmm.
However, like we discussed, for image recognition AI, it's a complete different beast. So these kind of AI, they're initially very early days. They are based on convoluted neural network or deep convoluted. I can't remember terminology, but I do understand what it is. It's a different species of AI trying to manage the signal on an image, right? So
Yeah.
for that, the system does not have the capability of communicating through human understandable language, so that it cannot tell you how it figures out, right?
How about to ask it, like asking it to mark it out, like the area of interest or something like that?
I still don't think it's gonna be happening that way.
Okay.
Yeah, because it's very simple. Let me make an analogy. It's basically like if you're using if you're using a typewriter, let's say, to type a letter, and then once the typewriter I mean, I'm so sorry, I cannot make any analogies. It's just maybe very hard to compare to anything else.
That's okay. My other question is, when I work with veterinarian pathologists, they're very much against the blinding. So they're saying it's almost impossible for them to
Right.
to diagnose, right?
Right.
So I'm just wondering whether AI, I mean, in this situation, is AI like complete blinded? They don't really know whether it's drug treated or something like that. So
I think that's a very, very important question. That's exactly what the AI should be, if improved much better than it is right now. I believe AI will outperform human. Explain a little bit. So veterinarian pathology is not much different than human pathologists like us, right? I totally agree, and I know exactly why that particular person claims that they are against the blindness, because what I just said, I earlier also talked about. It's called inter-observer variances, and we discussed how pathology is an imaging so-called science. And by the way, in the field ourselves, we always tend to call it art, meaning that it could have a different interpretation by different person. Almost like everybody looking at the famous picture of Mona Lisa, and everyone has a different interpretation. What's her facial expression? It's almost something like that. And we did also discuss that a HER2 plus in one pathologist's eye and might be HER2 just the 1 plus or even negative in a different pathologist's eye. So that's a very realistic existing issue that's also recorded and documented in many, many research publications, knowing that human observation opinion has a lot of variances. Not everything could be agreed upon. And even within one individual, say, if I am looking at a certain case at 6 or 7 p.m. Monday night, I'm so tired, I want to go home. I might say, oh, this is a benign. But then next day when I come back to the office, I'm energized, had a good sleep, had a good cup of coffee, and look at this case again. I'm like, oh my God, I didn't see this malignant cell. This is actually a malignant cancer. So that it's not uncommon in our field, in our role. I believe the same thing with a veterinarian pathologist as well. So it is a headache. It's a lot of explanation we have to sometimes deal with the FDA. We're trying to stand behind our conclusions of the study data. So thank you for mentioning that. But AI, I would say, could be much more objective. That's my conclusion.
no, thank you so much. Yeah, I agree, because I've often seen like different severity of histopathology findings, and they like it's almost hard to distinguish. It's like really arbitrary. So
Exactly.
and I mean there is a way called a peer review, but I guess similar in clinical pathology. I mean, for clinical setting too. So
that's true.
The other thing is, I mean, for ToxPath, they sometimes it's more like the committee or something, like they'll try to publish some kind of consensus for diagnosing specific findings, right? Like
Right.
the recommended consensus or something like that. I'm just wondering whether this is applicable to AI too. So
that actually goes back to the question I asked in terms of who's the gold standard, what's the ground truth. I can tell you some of the consensus could be eventually proven to be wrong, but that's the only way human can do these days, right? I don't know if I answered the question, but that's just my honest opinion against so-called consensus. Consensus, which helps to get you closer to the truth, but it does not mean it's actually the truth. It just means that most people will think this way. That's all the value of consensus. It doesn't mean the consensus is much closer to the truth than certain individuals believe. I don't know.
Yeah, I agree. It's always evolving, I guess.
Exactly. Yeah.
Thank you.
No problem. Thank you.
Dr. Song, I have a quick question about, first of all, thank you for all the presentation. For regular pathology, the immunohistochemistry usually use like 2, maximum 3 antibody staining one section. I agree with you. So you say if we use 3 or multiple antibodies staining the zero station, the section may be different. That's a current technology. Is it possible to use the primary antibody label different fluorescence staining one section?
Yes.
More than 3.
Yes. There is very mature. I can give
Okay.
you a recommendation. You can look into one is called iCura phenocycler fusion. Another one is called Lunaphore, Comet, COMET. Lunaphore spelled as LUNAPHORE. Those are pretty good systems.
Then usually like maximum roughly how many antibody can stain in one section?
I can speak for phenocycler fusion. You could do as much as up to 30 to 40 different antibodies on a single section with good quality, but I don't suggest go anywhere above that number. Although some publishers have said that they were successful to done 120 markers on a single section, which I believe you start to lose the accuracy of each marker staining if you crowded the markers on one single section.
Right. Yeah, for traditional immunohistochemistry, so if without secondary antibody, so usually not sensitive. But with AI, maybe more sensitive, can recognize the imager even without secondary.
Yeah, that's absolutely true. That's absolutely true. Because human eyes can only see certain wavelengths, and then it also depends on, remember we talked about what kind of a computer screen you're using, right? That makes a difference. And what kind of a camera sensor you are using in the scanner. I believe with the resolution and the sensitivity of the scanner, the camera in the scanners, it can pick up a lot of subtle lights that almost observable for human eyes, and that still make a difference, right? And also on the computer screen, this is our experience. We have been using computer detected signal. When we look at the images on the screen, it shows negative or pitch black, but actually when you put the cursor there to show the fluorescence activity, it's actually significantly higher than the background, meaning that your screen is probably just not expensive enough to reflect that differences.
Yeah, that's true. Okay. Yeah. Oh, can you repeat again for the website you recommend? What's the website?
Lunaphore, LUNAPHORE. PHORE. Yeah, I believe that's their Lunaphore is their company name, and their platform is called Comet, COMET.
COMET.
And then another one I strongly recommend is iCura, AKOYA.
AKOYA.
Yeah, that's the company's name, and the platform is called Phenocycler Fusion. PHENLCYCLER, Phenocycler Fusion, FUSION. By the way, since we're doing all this question and answer, just want to give a disclaimer, keep it as a record. I don't have any affiliations with any of these companies. So I'm not doing any commercials for any conflict of interest. I'm
Okay.
I'm users. I'm a user. Our company uses iCura's platform, but it's not the same as the one I just recommended, and we don't have any financial affiliations with them.
Okay.
That's fine.
Okay. Thank you very much. Appreciate
Of course. Thank you.
it. Okay.
Any other questions?
Yes. Hello.
Yes.
Thank you, Dr. Song. That's a general question regarding the people, who is not in our field, in not a clinical lab, in not pathology, pathologists or the pharmaceutical. They say in the future, the AI will be, the pathologists will be replaced by AI. So the pathology will be hard to find a job. What do you think?
I think it's exactly the same story for any other job on the on this planet, right? So if you think about what AI can do, maybe people think that even the surgeons might not be easily replaced by. I have a dot as well, because if you think about how AI is progressing, so it's a challenge for all different job description. But one thing I believe is for sure that um, being a pathologist myself, I'm not afraid of it replacing my job, but more I'm curious to learn how to be the master of AI, right? Rather being the slaves for AI. That's the most important. That means some challenges for people to continue learning, trying to understand more, trying to adapt to new technology. But well, I can only get to this level of an answer to you. Anything above that, I don't know either.
It's a good
Very
answer.
good.
Thank you.
Very good answer. I think this is a very good discussion. Any other questions from the audience? Then I ask the last questions. All these pathology, you look at that imaging is two-dimensional imaging. And when you do these paraffin or whatever these stuff, actually you can, the slides by slides, is that possible you can make the three-dimensional pathology studies? And
Yes,
do you think it is necessary to look at the third division about the current pathology studies? Should
that's
we add another division?
that's a great
It
question.
may, yeah.
It's actually happening. So if you search about 3D pathology imaging, and there are a lot of different ways, approaches to do that, and people are interested in 3D pathology, more, or how to say, the morphology. The reason being, especially for invasive cancer, right? Even after the surgery, you are a lot of time you worry about whether the tumor is completely taken out or there are residue tumors left in the body. And in this case, currently the most how to say, labor intensive way is that you just cut serial sections of the tissue block, especially at the edge of the surgical resection, and then you stain each one of them. And then right now the computer algorithm is mature enough to rebuild a 3D image or rendering of that particular piece. Tells you that where, if your marker represents the tumor, it can straightforwardly tell you where the tumor is infiltrating on the third dimension, not just the 2D planes. It's
Right.
in existence. It's just cost inhibitive for applying to actual clinical practice.
Okay. Thank you. Good. We have a very good discussion, and also, Dr. Song gives a tremendous, very decent talk tonight. I think everybody was very impressed. So let's thank you for his tremendous talk again. And now that our time is also up, so I just want to not only to appreciate Dr. Song's contribution for tonight's talk. Let me see. There's another. Let me see. Is there any other? Oh, there's no. Let me see. Any other question? Important for a med student who wants to. Oh, the comments. I think the people say it is important for a medical student who wants to go to pathology residency to match a hospital that has a state the art of technology and hospital that has technology, which hospital are advanced in technology? That's a question for the Dr. Song. Will the slides be shared? That two questions.
I can I probably have to modify the slides a little bit to make sure that there's anything that's you know, a borderline infringing anybody's copyright. And after that, I can definitely share a PDF copy of the slides.
Yeah,
That's the first question, easy question.
right.
The a little different, not different, but it's a little complex. That I can name a few pathology department in different hospitals in the U.S. that are pretty on the forefront of digital pathology. Um, for example, there's no ranking or there's no preference. I think University of Pittsburgh, UPMC Medical Center is where people are very interested in digital pathology. Ohio State is another center where a lot of activity. Ohio State, there's a Weizmann or Wayman Medical Cancer Center. You as a resident, you also get to go to do rotations there. Another one would be Harvard, but I can tell you, surprisingly, the Mass General, I don't think they're super technology advanced because it all depends on the funding, the money that Department of Pathology could have. But you can finish your residency, and most of the time, if you want to do digital pathology, you can pick a very advanced fellowship where Memorial Sloan Kettering is a good choice. MD Anderson, also very good choice. But I just have to warn you that there's no such a single medical center that has everything. They all have a little thing, and depends on they might be super rich, but still some of them might not be as highly advanced as a industrial, let's say, CRO company or focused company. So I guess I hope that answered your question. But at least some of these medical centers you can look into and look for the information yourself, and you can visit them, make your own choice.
Okay. So another question from audience is ask your connection way, how to connect with you. And I think that's in general, we will, today's talk will put in that we have in the same times, we not only use the Zoom meeting to have the audience to participate, and also that we also put that videotape in the YouTube, so you can find the YouTube again, if you want to look at that. And so all these connection information, all these things is also on that YouTube too. And also the other aspect is welcome everyone to join our the DC Great DC's uh, enterprise club. So as you are club member, and you can access the webpage to find all these the PPTs, videotapes, and all these things. We can do it. Okay. So now, as the time is up, and we also not only appreciate Dr. Song's talk tonight, we also appreciate the audience. The Saturday night and Saturday evening, you also still stay with us to learn more advanced technology and the progress in the AI, in the biotech and the pharmaceutical area. And here is next month's activity. And so our series talk now next one is the fifth talk. So now the fifth talk is basically talk about the AI application in healthcare and the regulatory compliance. So I hope that everyone join tonight. Next month you still can join us. And next month is in September 13th evening. And we will talk about the healthcare management and the regulatory compliance. So all with that, I thank you everyone and have a nice weekend. And we will continue that our series talk next month. So thank you very much. Bye-bye.
Thank you. Thank you all. Bye.
Thank you,
Thank you.
Dr. Zhang. Thank you, Dr. Yang.
Yeah, thank you.